Hey everyone, tonight we're going somewhere unexpected, not to a lost empire or a sunken continent, but inward, to the surface of your own body, to ask a question that turns out to have one of the most surprising answers in the whole of human science. How did some human beings become pale? Where did white skin come from? When did it arrive? And why? The answers involve ancient Africa, the physics of sunlight, a vitamin that turns out to be a hormone, a farming revolution nobody planned, and genetics that are still being worked out in laboratories right now. Before we begin, take a moment to subscribe, but only if you genuinely enjoy what I do here. And drop a comment telling me where in the world you're listening from and what time it is for you. Now dim the lights, get comfortable, and let's ease into tonight's journey together. Hold your hand up for a moment and look at it. Whatever shade you see, deep brown, warm tan, pale ivory, something in between, that colour is not decoration. It is not random. It is a document, a record written in the language of biology, of where your ancestors stood under the sky for thousands of years and what that sky demanded of their bodies. The science of human skin colour is one of the most illuminating stories in all of evolutionary biology, and it's also one of the most recently understood. For most of human history, we looked at skin colour and told stories about it. Stories about lineage and purity and destiny and hierarchy. The stories science has replaced those with are more complex, more surprising, and considerably more interesting. Our three companions tonight are drawn from three disciplines that together have cracked open this question in the last 2 decades. There is Dr. Amara, a geneticist at a research institute in Nairobi, who specializes in the evolutionary genomics of human pigmentation, the specific genes and mutations that control how much melanin a person's body produces and how those genes have changed over time. There is Professor Sven, a paleoanthropologist at a Scandinavian university who has spent 30 years studying the skeletal remains and genetic material of ancient Europeans, and whose work has fundamentally revised what we thought we knew about when and how quickly pale skin appeared on the continent. And there is Dr. Priya, a physiologist in Mumbai whose research focuses on the relationship between vitamin D, sunlight, and human health across populations, the biological mechanism that drove the whole transformation. Together they will take us through 11 chapters of one of the most personal stories that evolutionary biology has to tell. The question itself is deceptively simple. Most people, if asked, would say that white skin or pale skin is simply what happens when people live far from the equator for long enough. The sun is weaker in the north, so northern people became paler. This is broadly correct as a general principle, but spectacularly wrong as a specific account. The details, when it happened, how fast it happened, which genes changed, whether it happened the same way in Europe and East Asia, why some northern populations never developed pale skin at all, are far more complicated and far more revealing than the simple summary suggests. When Dr. Amara first encountered the ancient DNA evidence that rewrote the timeline of European skin colour, she found it almost impossible to believe. The data suggested that the people who'd been living in Europe for tens of thousands of years still had relatively dark skin well into what most people would think of as recent history. The transformation to pale skin, it turned out, was not the slow, gradual process that everyone had assumed. It was fast, shockingly fast, and understanding why requires going back to the very beginning. The human story begins in Africa, and for nearly all of its length, it stays there. Modern human beings, anatomically modern Homo sapiens with the same skeletal structure and brain capacity as people alive today, emerged in Africa somewhere between 200,000 and 300,000 years ago. The exact date is still being refined as new fossils are found and new dating techniques are applied, but the broad outline is clear. Africa is where we come from. Africa is where we spent the overwhelming majority of our existence as a species, and in Africa all human beings had dark skin. This was not a coincidence. It was the product of hundreds of thousands of years of natural selection operating in one of the most demanding solar environments on the planet. The equatorial regions of Africa, where our species emerged and where our ancestors spent the vast majority of their history, receive some of the most intense ultraviolet radiation anywhere on Earth. The sun is high in the sky year-round. Cloud cover is seasonal rather than constant. There is no deep winter to soften the light, and our ancestors, unlike almost all other primates, had lost most of their body hair, a development that probably preceded the appearance of our species, and that was connected to the evolution of efficient sweating as a cooling mechanism. Standing upright on the open savanna, hairless, the body exposed to the full force of equatorial sunlight, our ancestors needed protection. Melanin was that protection. Melanin is a pigment produced by specialized cells called melanocytes, which sit in the deepest layer of the skin. When ultraviolet radiation strikes the skin, melanocytes respond by producing melanin, which forms a kind of microscopic shield over the cell nucleus, absorbing the ultraviolet energy before it can penetrate to where the DNA is. Darker skin contains more melanin, distributed more densely and more efficiently, and provides dramatically better protection against ultraviolet damage than lighter skin. In equatorial Africa, this protection was not a cosmetic benefit. It was a survival necessity. The specific problem that melanin was protecting against is one that took scientists a surprisingly long time to fully appreciate. Ultraviolet radiation does not just cause sunburn and skin cancer, both of which are real concerns, but both of which typically operate on long timescales that are not the primary drivers of evolutionary selection. The more immediate problem is what ultraviolet radiation does to a vitamin called folate. Folate is a B vitamin that is essential for the synthesis and repair of DNA, and that is particularly critical during the rapid cell division of embryonic development. Without adequate folate, the neural tube, the structure that will develop into the brain and spinal cord, fails to close properly. Without adequate folate, pregnancies fail, children are born with severe developmental defects, and the reproductive success of the population declines. In the intense ultraviolet environment of equatorial Africa, dark skin was the difference between successful reproduction and reproductive failure. Natural selection does not negotiate with problems this fundamental. Dr. Amara emphasizes that the genetic evidence for this is unambiguous. When scientists analyze the genomes of different human populations and look at the genes that control melanin production, they find that the versions of these genes found in sub-Saharan African populations, the versions that produce high melanin levels, are also the most ancient. They are the versions closest to the ancestral state, the ones from which all other variants are derived. This is exactly what you would expect if dark skin is the original human condition and lighter skin is the derived, more recent state. The evolutionary story runs in only one direction. All human beings once had dark skin. Some populations later developed variants that reduced melanin production. The reverse, populations starting pale and developing dark skin, does not appear in the genetic record in any significant way. For roughly the first 200,000 years of our existence as a species, through the emergence of modern humans, through the development of sophisticated stone tools, through the beginnings of symbolic behavior and long-distance trade, and the other markers of what we call behavioral modernity, the entire human species was dark-skinned. There was no variation in this. There was no population of lighter-skinned people living somewhere on the margins. Dark skin was as universal a human characteristic as having 2 eyes or 5 fingers. It was simply what humans looked like. The transformation came later, and it came for reasons that are, once understood, both obvious and surprising. Around 60,000 years ago, possibly a little earlier, possibly a little later. The precise dating is one of the most intensely debated questions in paleoanthropology. A small group of human beings walked out of Africa, not all of humanity, not even a large fraction of humanity. A small group, numbering perhaps in the hundreds or very low thousands of individuals, crossed what is now the region between northeastern Africa and the Arabian Peninsula and entered a world that no modern human had ever seen. Everything that followed, the peopling of Asia, Australia, Europe, and eventually the Americas, descended from that initial crossing. The people who made this crossing carried with them the full suite of African adaptations that had made their ancestors successful on the continent. They were skilled hunters and gatherers. They could read landscapes and track animals and find water in difficult terrain. They had language and social structures and the accumulated practical knowledge of countless generations. And they had dark skin. The same dark, melanin-rich skin that had protected their ancestors under the African sun for hundreds of thousands of years. The route they took is still debated, but the most widely supported hypothesis involves a coastal path, following the shoreline of the Arabian Peninsula eastward, exploiting the rich marine resources of the coast, moving through what is now southern Asia, and eventually reaching Southeast Asia and Australia. The coastal route would have kept the migrants close to a reliable food source, and would have provided a navigational guide that required no maps or complex planning. Simply keep the water on one side and keep moving. Archaeological evidence, now substantially supplemented by ancient DNA analysis, is broadly consistent with this picture. Professor Sven has spent much of his career thinking about what these early migrants experienced as they moved into new environments. The coastline of the Arabian Peninsula 60,000 years ago was not the arid landscape it is today. During the glacial maximum, when global sea levels were lower and more water was locked up in ice sheets, large parts of the Arabian interior were wetter and more hospitable than they are now. But as the migrants moved north and east into Central Asia and eventually toward Europe, the environment began to change in ways that would eventually have profound consequences for their descendants. The sky changed. The sun, which had beaten down with equatorial ferocity in Africa, began to follow a different path, lower in the sky, weaker and in winter, present for only a few hours each day. The ultraviolet radiation that had been so abundant in Africa, and that the dark skin of the migrants had evolved to manage, became scarce. And scarcity, it turned out, was a problem just as serious as excess. The migration was not a single journey. It was a multi-generational process, a slow expansion across decades and centuries and millennia, with each generation pushing a little further into new territory, establishing new camps that became the base for the next wave of exploration. Individual people did not walk from Africa to Europe, but their descendants, over 50,000 years, filled the world, and with each degree of latitude gained, with each 100 kilometers moved north, the selective pressure on their skin was quietly, inexorably changing. Here is the problem that pale skin solved, and it is important to understand it clearly because it is the engine that drove the entire transformation. Vitamin D is not technically a vitamin at all. It behaves like a hormone, a chemical messenger that regulates dozens of physiological processes throughout the body. It is produced in the skin when ultraviolet B radiation triggers a chemical reaction that converts a cholesterol precursor into the active form of the molecule, and in the absence of adequate ultraviolet B radiation, the body cannot make enough of it. The consequences of vitamin D deficiency are severe and wide-ranging. The most visible and historically most significant is its effect on bone development. Vitamin D is essential for the absorption of calcium from the gut and the deposition of calcium into bone tissue. Without it, bones fail to mineralize properly. In children, this produces a condition called rickets. bones that are soft enough to bend under the weight of the body, producing the characteristic bowed legs and deformed spine and ribcage that ancient skeletal remains reveal with disturbing frequency. In adults, the equivalent condition, osteomalacia, causes bones to lose mineral density, becoming fragile and prone to fracture from ordinary activities. Dr. Priya has devoted her career to understanding the downstream consequences of vitamin D deficiency, and she's emphatic about one aspect of the problem that is often underappreciated. The most evolutionarily significant effect of vitamin D deficiency is not on the individuals who suffer from it, but on their ability to reproduce. In women, childhood vitamin D deficiency that causes even subtle pelvic deformation can make childbirth catastrophically dangerous. The birth canal, shaped by bones that did not develop properly due to calcium deficiency, may be too narrow for a full-term infant to pass through. Before the advent of modern obstetric surgery, this was a death sentence, for the mother, for the infant, or for both. In a world where childbirth mortality was already high, this additional risk was devastating. Now imagine this pressure operating on a population of dark-skinned people who have recently moved into Northern Europe. The ultraviolet radiation at high northern latitudes is dramatically weaker than at the equator, not just in winter when the sun barely rises above the horizon, but even in summer, when the angle of the sun means that ultraviolet rays are passing through much more atmosphere before reaching the ground. For a person with dark skin, which evolved to block most ultraviolet radiation, the situation is acute. The skin is doing exactly what it evolved to do, protecting against ultraviolet damage, but in an environment where ultraviolet is scarce rather than abundant, this protective function has become a liability. The melanin that was essential in Africa is now blocking the very radiation that the body needs to produce vitamin D. The mathematics of this situation are stark. Dark skin can reduce ultraviolet transmission through the skin by 90% or more. In Africa, this is appropriate. There is so much ultraviolet available that even with 90% blocked, the remaining 10% is sufficient for adequate vitamin D synthesis. In Northern Europe, where ultraviolet levels might be only 10 or 15% of what they are in equatorial Africa, the same 90% reduction leaves the skin with access to only 1 or 2% of the ultraviolet needed for vitamin D production. This is not enough, not even close to enough. The result is chronic severe vitamin D deficiency, the kind that deforms children's bones, weakens immune systems, and kills women in childbirth. Dr. Priya is careful to point out that this was not a sudden crisis. The populations that moved into northern latitudes faced this problem gradually, over many generations, as they pushed further and further from the equator. Some behavioral adaptations were probably tried, spending more time outdoors in summer, removing clothing when the sun was out, settling preferentially in more southerly or sunnier locations. But behavior can only compensate so much. The laws of photochemistry do not respond to cultural workarounds. What was needed was a biological solution, a body that could capture more ultraviolet radiation from a weaker sun, and that required different skin. Natural selection works through a simple mechanism. Random mutations occur constantly in every genome, in every generation. Most of these mutations have no significant effect on survival or reproduction. They accumulate neutrally in the population, neither helped nor hindered by selection. But occasionally, rarely but inevitably, a mutation occurs that changes something important. That changes how well an organism survives or reproduces in its particular environment. If the change is beneficial, the individuals who carry it have more surviving offspring than those who do not. Over generations, the beneficial mutation spreads through the population. The speed at which it spreads depends on how large the survival advantage is. In the populations of dark-skinned people living in northern latitudes with chronic vitamin D deficiency, any mutation that reduced melanin production would have provided a survival advantage. The reduction would not need to be dramatic. Even a modest reduction in melanin levels would translate into modestly better vitamin D synthesis, which would translate into modestly better bone development, modestly better immune function, and slightly improved survival of mothers and infants through childbirth. Over many generations, even a small survival advantage causes a genetic variant to spread through a population. The specific mutations that produced the lighter skin of modern Europeans have been identified with considerable precision by modern genetic analysis. The most significant, in terms of its contribution to lighter skin in European populations, is a variant in a gene called SLC24A5. This gene encodes a protein involved in the production of melanin, specifically in the transport of ions across the membrane of the melanosomes, the tiny organelles within melanocytes where melanin is synthesized. The ancestral version of this gene, found in nearly all Africans and in most other non-European populations, produces a protein that supports high melanin production. A single change, one nucleotide in the gene sequence, produces an altered protein that is less efficient, and the result is significantly reduced melanin production and lighter skin. Dr. Amara finds the SLC24A5 story almost philosophically striking. One letter in a sequence of roughly 3 billion, a single typographical error in an encyclopedia the size of a building. And that error, copied faithfully into every cell of every person who carries it, changes the appearance of their skin enough to be immediately visible. The mutation is present in nearly all people of European ancestry today, and is essentially absent in sub-Saharan African populations. Its prevalence in Europe is not the result of genetic drift, the random fluctuation of gene frequencies in small populations. Its frequency is too high, and its spread too rapid to be explained by chance. The signature of strong positive selection is unmistakable. This mutation was actively favored by natural selection because it helped people survive. But SLC24A5 is not the only gene involved, and the genetics of skin color are considerably more complex than a single mutation story would suggest. Multiple genes contribute to skin pigmentation, each with its own history of mutation and selection. SLC45A2 is another gene whose variant is strongly associated with lighter skin in European populations. Variants in genes affecting the production and type of melanin, the relative proportions of the darker eumelanin and the redder pheomelanin, contribute to the full range of pigmentation from pale to dark. The analysis of ancient DNA has allowed scientists to track when each of these variants appeared in European populations and how quickly they spread. Professor Sven was involved in some of the early ancient DNA studies that revealed the timeline of skin lightening in Europe, and he describes the moment when the results first became clear as one of the most memorable of his scientific career. The ancient European hunter-gatherers, the people who had been living in Europe for tens of thousands of years, the direct descendants of the first modern humans to colonize the continent, still carried the ancestral dark-skin variants of these genes. Their genomes, extracted from skeletal remains and analyzed in the laboratory, showed that as recently as 8,000 or 9,000 years ago, the people living in Britain and Scandinavia and Central Europe still carried the genes for relatively dark skin. The rapid shift to the light skin variants that are now nearly universal in European populations happened very recently, within the last 10,000 years, and in some cases perhaps within the last 6,000 or 7,000 years. The timeline of skin lightening in Europe presents a puzzle. Modern humans had been living in Europe for 40,000 years before the rapid shift to lighter skin occurred. If the vitamin D pressure was the driver, why did it take so long? Why didn't lighter skin evolve as soon as people arrived in northern latitudes? The answer lies in a transformation that reshaped human life in ways that went far beyond agriculture itself, and that had a profound and unexpected effect on the vitamin D equation. For the 40,000-year period before agriculture, the people living in northern Europe were hunter-gatherers. They lived by hunting large and small game, fishing and collecting wild plants. This lifestyle, demanding and uncertain as it was, had one nutritional advantage that is easy to overlook. It provided access to some of the richest dietary sources of vitamin D available. Fatty fish, salmon, herring, mackerel, eel, are among the most vitamin D-dense foods on Earth. Animal livers, particularly from large game, contain substantial amounts. Shellfish and other marine foods contribute significantly. A hunter-gatherer diet in northern Europe, especially one that included regular access to coastal or riverine resources, could provide enough dietary vitamin D to compensate significantly for the limited ultraviolet synthesis that dark skin would achieve under a northern sun. This is why, Dr. Priya explains, the hunter-gatherers of ancient Europe could persist with relatively dark skin for so long in a low ultraviolet environment. They were eating their vitamin D rather than synthesizing it. The selective pressure for lighter skin was present but was being partially offset by dietary compensation. The vitamin D crisis was real but manageable. The situation changed dramatically when agriculture arrived. Agriculture came to Europe from the Middle East, brought by farming populations who had domesticated wheat and barley in the Fertile Crescent, and who expanded westward and northward over several thousand years, beginning around 9,000 years ago. The agricultural transition was not simply a change in subsistence strategy. It was a revolution in diet, in settlement pattern, in social organization, and ultimately in biology. And in one critical respect, it made the vitamin D situation dramatically worse. Early agriculture was based on cultivated grains, wheat, barley, millet, rye. These grains are nutritionally dense and calorically rich, which is why they became the foundation of human diet for the next 10,000 years. But they contain essentially no vitamin D. Plants do not produce vitamin D. Animals do in specific tissues, particularly fat and liver. A diet based primarily on grain has almost no vitamin D content. The shift from a varied hunter-gatherer diet, rich in fish, meat, and wild animal products, to an agricultural diet based primarily on cultivated grains removed the dietary vitamin D that had been compensating for limited skin synthesis. Suddenly, the populations of northern Europe were in a true vitamin D crisis. Not the manageable, partially compensated deficit of the hunter-gatherer period, but a severe, uncompensated deficiency that was producing epidemic levels of rickets in children and catastrophic rates of birth complications in women. The selective pressure for lighter skin intensified enormously, not just because of the northern latitude and its weak sunlight, but because the dietary safety net had been removed. Professor Sven has excavated dozens of sites from the early agricultural period in northern Europe, and the skeletal evidence is striking. Ricket signatures, the telltale bone deformities that result from childhood vitamin D deficiency, are far more common in early agricultural communities than in the hunter-gatherer populations that preceded them. The farmers were shorter, more prone to disease, showed more signs of nutritional stress. The transition to agriculture was, in the short term, a health disaster, and within that disaster, the selection pressure for lighter skin became intense enough to drive the rapid genetic transformation that the ancient DNA record reveals. The light skin variants that had been rare in hunter-gatherer populations swept through the agricultural communities with extraordinary speed, exactly what you would expect when the survival advantage of a genetic variant becomes very large very quickly. The agricultural revolution accelerated skin lightening in another way too. Farmers are sedentary in a way that hunter-gatherers are not. They stay in one place, working their fields, storing their grain, building their villages. This means they spend considerably more time indoors, in longhouses and storage buildings and workshops, than hunter-gatherers who are constantly moving through the landscape. Less time outdoors means less skin exposed to whatever sunlight is available. Less sun exposure means less vitamin D synthesis. The farmer's lifestyle compounded the farmer's diet to create a perfect storm of vitamin D deficiency that would only be resolved when the genetic variants for lighter skin had spread through the population. One of the most striking findings of modern genetics is that pale skin did not evolve once and then spread around the world. It evolved independently through different genetic mutations in different populations facing similar environmental pressures. This is a phenomenon called convergent evolution, the same adaptive outcome arising through different genetic mechanisms in different lineages. It tells us something profound about the relationship between environment and biology. The European story, which we've been tracing through the SLC24F5 mutation and its relatives, is one version of this tale. But East Asian populations also developed lighter skin than equatorial Africans, and they did so through largely different genes. The genetic variants that lightened skin in East Asian populations, particularly variants in genes such as OCA2 and others that are still being characterized, are almost entirely different from the European variants. A European and an East Asian person of similar skin tone have that skin tone for different genetic reasons, through different mutations that found different molecular pathways to the same destination. Dr. Amara finds this fact one of the most instructive in all of human evolutionary genetics. It is a clear demonstration that natural selection was responding to a real and significant environmental pressure. The pressure was strong enough to produce similar outcomes in multiple independent lineages. If lighter skin in northern latitudes was simply a genetic accident, a mutation that drifted to high frequency by chance, you would not expect to see it evolve independently in separate populations. The fact that it did is strong evidence that the selection pressure was real and powerful. The timing of skin lightening in East Asian populations appears to be similar to the European timeline, mostly within the last 15 to 20,000 years, with some variants spreading particularly rapidly during and after the transition to agriculture in East Asia around 9 to 10,000 years ago. The parallel with the European story is remarkable. Different populations, different continents, different crops, different genetic mechanisms, but the same basic dynamic. Agriculture removes dietary vitamin D. Northern latitudes provide weak sunlight. Dark skin blocks what little ultraviolet is available. The body needs lighter skin to survive. Selection rapidly favors the mutations that provide it. Not all northern populations developed pale skin, and the exceptions are as informative as the examples. The indigenous peoples of the Arctic, populations who have lived for thousands of years at some of the highest latitudes on Earth, where winter brings weeks of complete darkness, are not the palest people on the planet. Traditional Arctic populations maintained moderate to relatively dark skin despite their extreme northern location. This apparent paradox dissolves when you look at their traditional diet. Arctic peoples subsisted primarily on marine mammals, seals, walruses, whales, and fish. These foods are extraordinarily rich in vitamin D. A single meal of seal liver can provide more vitamin D than most people would synthesize in weeks of sun exposure. When dietary vitamin D is abundant, there is no selection pressure for lighter skin. The Arctic people solved the vitamin D problem through diet rather than through pigmentation, and their skin reflects this. Darker than would be expected from their latitude because the latitude was never the real problem. Professor Sven finds this comparison illuminating in both directions. The Arctic exception proves the rule. It confirms that the selective force driving skin lightening was not simply latitude or cold or any other correlate of northern environments, but specifically the availability of vitamin D. whether through synthesis or diet. The populations that lacked dietary sources of vitamin D were the ones whose skin lightened most rapidly. The populations that had abundant dietary sources could maintain darker skin without suffering the consequences. Skin color is the most visible and the most extensively studied of the human traits that vary across populations, but it is not the only one. Several other characteristics tend to cluster with lighter skin in northern populations, and understanding their origins adds texture and complexity to the overall story. Blue eyes are perhaps the most striking of these associated traits. The iris of the eye, like the skin, contains melanin, the same brown-black pigment that darkens skin. Dark brown or nearly black eyes, which are the ancestral human condition, result from high concentrations of melanin in the front layers of the iris. Blue eyes result not from blue pigment but from a reduction in melanin concentration. When there is little melanin in the iris, light entering the eye is scattered in a way that produces the perception of blue, the same physical phenomenon that makes the sky blue. It is an optical effect, not a pigment. The genetic change responsible for blue eyes in European populations has been traced to a specific variant in a region near the OCA2 gene, a gene that also influences skin color. This has led to the hypothesis that eye color and skin color lighten together, at least in part, as a consequence of the same selective pressure. If variants that reduced OCA2 activity spread because they lightened skin and improved vitamin D synthesis, the same variants may have incidentally reduced iris melanin as a side effect. Blue eyes, on this view, might be partly a byproduct of selection for lighter skin rather than an independently selected trait. However, the timing is complicated. Genetic evidence suggests that the blue eye variant in European populations arose earlier than the major skin lightening variants, perhaps 20 or 30,000 years ago, compared to the 10 to 15,000 years estimated for the key skin variants. This has led some researchers to propose that blue eyes were favored independently, perhaps through sexual selection, the phenomenon by which traits become common not because they improve survival, but because they are preferred by potential mates. In small, familiar communities where everyone knows everyone, an unusual eye color might stand out and become associated with desirability. If blue-eyed individuals attracted more mates and had more children, the trait would spread even in the absence of any survival advantage. Dr. Amar is candid about the limits of current understanding here. The genetics of eye color are better characterized than those of many other traits, but the question of why blue eyes evolved, survival advantage, sexual selection, or genetic hitchhiking on skin color variants, has not been definitively resolved. What is clear is that blue eyes are recent, that they arose in Europe, and that they spread rapidly enough to suggest either strong selection or a fortunate combination of selection and drift in the populations where they first appeared. Hair color follows a similar story. The range of hair colors seen in European populations, from very dark brown or black through various shades of brown to blonde and red, represents variation in the same melanin system that controls skin color. Blonde hair results from reduced overall melanin production. Red hair results from a shift in the balance between eumelanin, the dark pigment, and pheomelanin, the red-orange pigment, due to variants in a gene called MC1R. The selection pressures on hair color are less well understood than those on skin color, partly because hair covers a smaller surface area and contributes less to whole-body vitamin D synthesis. Body proportions show another set of trends that are related to climate but to temperature rather than sunlight. Human populations that evolved in cold environments tend, on average, to have shorter limbs relative to body size, more compact and stockier builds, and different distributions of body fat than populations from warmer regions. These differences follow patterns that engineers recognize from basic thermodynamics. A compact shape with a low surface area-to-volume ratio retains heat more efficiently than an elongated shape with a high ratio. The longer limbs of populations from hot climates function as radiators, dissipating heat. The shorter limbs of cold climate populations reduce heat loss. Natural selection shaped body form to match the thermal environment, just as it shaped skin to match the solar environment. For the vast majority of human evolutionary history, the relationship between skin color and geography was tight. People lived where their ancestors had lived. The sun they were born under was the same sun that had shaped the pigmentation genes they carried. Dark-skinned people lived near the equator, where dark skin was needed. Lighter-skinned people lived at higher latitudes, where lighter skin was advantageous. The evolutionary logic was locally coherent. Then, in a geological instant, everything changed. The last 5 centuries of human history have involved the most rapid and extensive movement of people across the planet that our species has ever experienced. Colonization, the slave trade, voluntary migration, refugee movements, economic migration. These processes have transplanted people from every genetic background to every latitude, creating populations whose inherited pigmentation often has no relationship to the sunlight they actually live under. The evolutionary calibration that took tens of thousands of years to establish has been scrambled in a historical blink. Dr. Priya studies the health consequences of this mismatch, and her research reveals a pattern of misaligned adaptation that plays out in both directions. When people with very dark skin that evolved to manage intense tropical ultraviolet radiation, live at high northern latitudes where ultraviolet is scarce, the same protection that was essential in their ancestral environment becomes a barrier. The melanin that shields folate from ultraviolet destruction also blocks the synthesis of vitamin D. In a low ultraviolet environment, dark skin cannot capture enough of the available radiation to produce adequate vitamin D. The result, well documented in the clinical literature, is elevated rates of vitamin D deficiency in dark-skinned populations living at high latitudes, with downstream effects on bone health, immune function, mood, and several other physiological systems. The reverse situation creates different but equally real health challenges. People with very light skin that evolved to maximize ultraviolet capture in low-light northern environments, who live in high ultraviolet tropical or subtropical environments, are exposed to radiation levels that their melanin was never designed to manage. The protective barrier is too thin. Ultraviolet penetrates too deeply. DNA damage accumulates in skin cells. The result is dramatically elevated rates of skin cancer, melanoma, basal cell carcinoma, squamous cell carcinoma, in fair-skinned populations living in high ultraviolet regions. Pale-skinned populations in Australia, South Africa, and similar environments experience some of the highest skin cancer rates ever recorded. What makes the modern situation particularly interesting, Dr. Priya notes, is that human beings are now using technology to do what evolution did slowly. Sunscreen blocks ultraviolet radiation, providing artificial protection that substitutes for melanin. Vitamin D supplements compensate for the vitamin D that skin cannot synthesize in low ultraviolet environments. These interventions are imperfect. Sunscreen is applied inconsistently, supplements are taken irregularly, and neither replaces the nuanced, continuous automatic response of melanin in the skin. But they represent a fundamentally new kind of adaptation, cultural and technological rather than biological. For the first time in the history of our species, we can live, at least tolerably well, under any sky regardless of the skin we were born with. Whether evolution is still acting on skin color in modern human populations is a question that researchers approach with caution. The answer is probably yes. Natural selection never stops entirely, but the strength and direction of selection has been profoundly altered by medicine, technology, and global movement. The tight link between latitude and pigmentation that drove rapid change in the past is now disrupted by interventions that short-circuit the biological consequences of the mismatch. Selection for lighter skin in northern populations was so rapid and so powerful because vitamin D deficiency killed people before they could reproduce. Modern medicine prevents those deaths. Selection for darker skin in high ultraviolet environments was powerful because ultraviolet damage caused cancer and folate destruction that reduced reproductive success. Sunscreen and supplementation modify those pressures. Evolution continues, but it is responding to a transformed landscape of risks and advantages. When you understand the story of how human skin color evolved, when you trace it from its origins in the ultraviolet environment of equatorial Africa, through the vitamin D pressures of northern latitudes, through the agricultural revolution, through the independent parallel evolution of similar solutions in separate populations, a particular understanding settles into place. Skin color is the solution to a problem, not one problem, but a set of related problems involving ultraviolet radiation and its consequences, both beneficial and harmful, for the human body. It is a physiological adaptation, as specific and as functional as any other feature of human anatomy. This understanding has implications that are worth dwelling on. The genetic variants that produce different skin tones are not randomly distributed across the genome. They are concentrated in a small number of genes, all of which are involved in the melanin production pathway. The rest of the human genome, the vast majority of human genetic variation, shows no meaningful correlation with the skin color variations that we have built so much cultural and social architecture around. Two people with very similar skin colors may be more genetically different from each other than either is from someone with very different skin color, because skin color is controlled by a handful of genes while overall genetic similarity depends on the full genome. Professor Sven returns to this point when he thinks about the ancient DNA work that has characterized his career. The ancient Europeans whose genomes he has helped to sequence were not the same people, genetically, as modern Europeans. They were dramatically different in ancestry, in the frequencies of various genetic variants, in their relationships to other populations. Skin color is one of the few aspects of their appearance that the genetic record allows us to reconstruct with confidence. But the reconstruction consistently shows that skin color changed faster, and more recently, than almost any other visible feature. The people living in Northern Europe 8,000 years ago looked, in terms of skin color, more like people who live today near the equator than like the pale-skinned populations that now occupy the same latitudes. The connection between northern latitude and pale skin is real, but it's also recent, far more recent than most people assume. Dr. Amara thinks about this in terms of what it says about the nature of human diversity more broadly. Skin color is among the most visible ways that human beings differ from one another. It is immediately apparent, it is associated with large geographic patterns, and it is the characteristic around which more cultural meaning has been built than almost any other biological trait. And yet it is, in the grand scheme of human genetic variation, a relatively superficial difference. The genes involved are few. The evolutionary history is short. The adaptive logic is straightforward. The variation that exists within human populations is far greater in terms of total genetic diversity than the variation between populations defined by skin color. The scientific understanding of skin color does not erase the cultural and social significance that human societies have attached to it. That significance is real in its effects on people's lives, whatever its biological basis or lack thereof. But it does clarify what the biological story actually is. It is a story about ultraviolet radiation and vitamin D and folate and the astonishing flexibility of the human body in responding to different environments. It is a story about how quickly a species can change its appearance when the survival pressure is strong enough. It is a story about convergent evolution, the same solutions arising independently in separate populations because the problems they faced were the same. It is not a story about hierarchies or categories or essential differences. It is a story about one species adapting over and over again to the particular slice of the planet it happened to occupy. We have travelled a long way tonight, from the open savannas of equatorial Africa to the dark winters of Ice Age Europe, from the fishing shores of ancient Scandinavia to the grain fields of the Neolithic Middle East. We have followed the trail of a small number of genetic mutations as they spread through human populations over thousands of years, responding to the specific demands of specific environments. We have watched skin colour change, faster than almost anyone expected, through mechanisms more varied and more recently understood than the textbooks of a generation ago suggested. Dr. Amara sits in her laboratory in Nairobi, looking at the data from a new set of ancient genomes that her team has just finished analysing. The samples are from a region of East Africa where the evidence suggests modern humans may have been particularly ancient, the population from which all subsequent human expansion ultimately traces its origin. The pigmentation gene variants in these genomes are exactly what she would predict. The ancestral forms, high in melanin, the original human software unchanged by the pressures that would later reshape it in populations far from home. She finds this continuity moving in a scientific way. The beginning of the story is still legible in the genomes of people whose ancestors never left. Professor Sven is in Stockholm, reading proofs of a paper that has taken 3 years to write. A comprehensive analysis of skin colour genetics in ancient European populations across a span of 30,000 years, from the earliest modern human arrivals to the medieval period. The paper's central finding, which he has spent much of his career building the evidence for, is the extraordinary recentness of the European pigmentation shift. The people who walked into Northern Europe 40,000 years ago had skin that the ancient DNA record says was considerably darker than what we would associate with Scandinavia today. The transformation to the pale skin that now characterizes Northern European populations was a process of the Neolithic and Bronze Age, the last 10,000 years at most, and possibly considerably less. Dr. Priya is seeing patients this evening, a mix of people whose ancestral origins and current environments are mismatched in various ways. People dealing with vitamin D deficiency, or with the dermatological consequences of ultraviolet exposure for which their skin was not calibrated. She explains to each of them, patiently and in whatever language of complexity is appropriate, that their skin was optimized for somewhere that is not where they are now, and that this is a solvable problem if they understand what is happening. She finds that when people understand the evolutionary logic, when they understand that their skin color is a historical record of where their ancestors lived, not a marker of identity or worth, they relate to their own biology differently, with more curiosity, she finds, with more interest, with something that looks, sometimes, like relief. Your skin, whatever its color, is a document. It records something real. The ultraviolet environment that your ancestors inhabited, the pressures that shape their biology, the solutions that natural selection arrived at over thousands of years in specific places under specific skies. It is a document of adaptation, not of hierarchy, a record of where your people stood under the sun and what the sun asked of them and how their bodies answered. Look at your hand one more time. The colour you see is the colour of a particular evolutionary history, a history that began in Africa, that branched and travelled and adapted and reconverged, that is still being written in every generation as people move and mix and carry their genetic histories into new environments. The story is not finished. It has never been finished. But tonight, in this quiet hour before sleep, you know more of it than you did before, and that seems like enough to carry into the dark. This has been Midnight Chronicle. Sleep well, wherever in the world you are tonight. The African continent is not a single environment. It spans an extraordinary range of climates and habitats, from the Sahara in the north to the tropical rainforests of the Congo Basin to the temperate grasslands of the south. But the region where modern humans appear to have emerged, and where the earliest fossils and genetic signals point, is in the savanna and semi-arid zones of eastern and southern Africa, environments characterized by intense year-round sunlight and relatively open terrain. It was in these conditions that the relationship between human skin and ultraviolet radiation was first established, and it was these conditions that produced the dark pigmentation that is the ancestral human state. The mechanism by which natural selection maintains dark skin in high ultraviolet environments is not just about blocking damage. It is also about preserving a specific molecule that is essential for reproduction. Folate, the vitamin whose destruction by ultraviolet radiation drove the initial evolution of dark skin, is not just important for fetal development. It is important throughout life, for the synthesis and repair of DNA in all rapidly dividing cells. The hair follicle, the lining of the gut, the immune system, all of these systems depend on continuous folate availability. But the most acute, most evolutionarily significant requirement for folate is during the earliest weeks of fetal development, when the neural tube closes and the foundations of the nervous system are laid. A brief, severe folate deficiency at this critical window can result in neural tube defects that are incompatible with survival. For a pregnant woman spending her days in equatorial sunlight with insufficient melanin protection, this risk was not theoretical. It was a constant biological reality. Dr. Amara points to an interesting natural experiment that illustrates this relationship. Studies of human populations living at high altitudes in tropical regions, such as the highlands of Ethiopia or the Andean populations of South America, show that they maintain very dark skin despite temperatures that might seem more appropriate to northern climates. The key variable is not temperature, but ultraviolet radiation. At high altitude, the atmosphere is thinner and filters out less ultraviolet, so the radiation reaching the skin is actually more intense than at sea level, even if the air is cooler. The skin is adapted to the ultraviolet environment, not the thermal one. This is a clear demonstration that melanin is responding to light, not to heat, and that the relevant evolutionary pressure is photochemical rather than thermodynamic. The genetic architecture of dark skin is itself instructive. When geneticists look at the specific variants in pigmentation genes that are most common in sub-Saharan African populations, they find evidence of strong positive selection, the statistical signature of variants that were actively favored by natural selection rather than simply drifting to high frequency by chance. This tells us that dark skin was not simply neutral, not simply the default state that happened to persist because there was no pressure to change it. It was actively maintained by selection. Any mutation that reduced melanin production in the ancestral African environment was selected against. It reduced survival and reproduction, and it was eliminated from the gene pool. Dark skin was not just the starting point. It was a competitively maintained adaptation that was actively preserved for hundreds of thousands of years. The question of why it took so long for the first modern humans to leave Africa is one of the more intriguing puzzles in paleoanthropology. Modern humans were anatomically present in Africa by at least 200,000 years ago, and there is growing evidence for behavioral modernity. The use of ochre for pigmentation, the production of geometric and potentially symbolic engravings, long-distance transport of particular raw materials from dates approaching this age. Yet the successful colonization of the world outside Africa appears to have happened much later, around 60,000 years ago. with perhaps some earlier attempts that did not result in lasting populations. Something kept humans in Africa or prevented successful establishment outside it for 100,000 years or more. Various explanations have been proposed. Climate instability during the period from roughly 200,000 to 70,000 years ago, including the dramatic bottleneck event that may have reduced human populations to dangerously low levels around 70,000 years ago, may have prevented the accumulation of the cultural and technological complexity needed for successful long-distance migration. The Neanderthal populations already established in Europe and Western Asia may have been effective competitors that prevented modern human expansion into those territories. The crossing from Africa to Arabia may have required specific climatic conditions: a greened Arabian Peninsula, lower sea levels, reduced width of the Red Sea crossing, that only occurred episodically. Most likely all of these factors played roles, and the migration that eventually succeeded was the result of a particular convergence of circumstances rather than a single cause. When the migration did succeed, the speed of subsequent expansion was remarkable. From the initial crossing point, modern humans spread across southern Asia and reached Australia within a few thousand years, a pace of expansion that implies active exploration and rapid exploitation of new territories rather than a slow, hesitant diffusion. The people making this expansion were not cautious or timid. They were skilled, adaptable, and clearly willing to move into completely unknown environments. Archaeological sites from this period in Australia and Southeast Asia show the immediate development of new technologies adapted to local conditions, new types of tools, new approaches to resource exploitation, new forms of social organization appropriate to new environments. The first people to reach Australia were not struggling refugees. They were confident colonizers of an empty continent. Professor Sven emphasizes that the genetic evidence from living populations around the world allows us to trace this expansion with increasing precision. The genetic diversity of living populations decreases systematically the further they are from Africa, a consequence of the serial founder effects that occur when small groups break off from larger populations and move into new territories. Each new population is founded by a subset of the previous one, carrying only a fraction of the genetic diversity. This declining diversity pattern, visible across thousands of genetic markers, is one of the strongest lines of evidence that all non-African populations are descended from a relatively small group that left Africa and expanded, with each successive wave of migration reducing genetic diversity further. The genes that these founding populations carried, including the ancestral dark-skin variants, went with them into every new environment they colonized. The physiology of vitamin D production is worth understanding in some detail, because the details illuminate why skin color matters so much to vitamin D status. When ultraviolet B radiation, specifically the range of wavelengths between 290 and 315 nanometers, strikes the skin, it converts a molecule called 7-dehydrocholesterol, which is present in skin cells, into previtamin D3. This previtamin D3 then undergoes a thermal isomerization, a heat-driven molecular rearrangement, to become vitamin D3. This initial skin synthesis step produces an inactive form of the vitamin. It is then transported to the liver, where it is converted to a partially active form called 25-hydroxyvitamin D, and then to the kidney, where a final conversion step produces the fully active form called calcitriol, which is the molecule that acts as a hormone throughout the body. The critical point in this chain, from the perspective of skin color and latitude, is the initial step, the conversion of 7-dehydrocholesterol in the skin. This step requires ultraviolet B radiation at specific wavelengths, and the amount that reaches the skin depends on two independent factors: the intensity of ultraviolet B in the incoming sunlight, and the amount of melanin in the skin that absorbs or scatters the ultraviolet before it can reach the deeper skin layers where the 7-dihydrocholesterol is located. Both factors can limit vitamin D synthesis, and both vary in ways that create the distinctive geographic pattern of skin colour that characterises our species. Dr. Priya has spent years measuring vitamin D status in populations with different skin colours living at different latitudes, and her data confirm the basic picture that the evolutionary story predicts. At the equator, both dark-skinned and light-skinned people typically have adequate vitamin D status. There is enough ultraviolet B to support synthesis even through heavy melanin protection. At high latitudes, the differences become striking. In winter months in northern Europe or northern North America, people with very dark skin often have vitamin D levels that are clinically deficient, below the threshold associated with bone health effects and immune dysfunction. People with lighter skin maintain higher levels, though even they can become deficient during long northern winters with limited outdoor exposure. The seasonal pattern of vitamin D synthesis is itself a crucial part of the story. At high latitudes, ultraviolet B radiation is adequate for vitamin D synthesis for only part of the year, roughly from April to September, at latitudes around 50 degrees north, and for an even shorter window further north. During the rest of the year, even full skin exposure to midday sunlight cannot produce significant vitamin D, because the sun is too low in the sky and the ultraviolet B is filtered out by the thick column of atmosphere the light must traverse at low sun angles. This means that even with light skin, northern populations must accumulate enough vitamin D during the summer months to sustain them through the winter, a biological storage problem that the body addresses by depositing vitamin D in fat tissue, but that still creates seasonal swings in vitamin D status that can be medically significant. The ancient DNA revolution that has transformed our understanding of human evolution over the past decade has been particularly illuminating for the question of skin colour, because pigmentation genes are among the traits whose genetic basis is well enough understood that we can infer appearance from genotype with reasonable confidence. When researchers extract and sequence ancient DNA from skeletal remains, they can look at the specific variants present in the pigmentation genes and make probabilistic predictions about the skin colour of the individual. This is not perfect. We do not yet understand all the genes involved in pigmentation, and there is always uncertainty in the relationship between genotype and phenotype. But for the major pigmentation genes that are well characterised, the predictions are reliable enough to tell a coherent story. The story that ancient DNA tells about European skin colour is one of the most surprising findings in recent human evolutionary research. The earliest modern humans in Europe, the people who arrived around 45,000 years ago during the period known as the Upper Paleolithic, carried pigmentation gene variants that predict dark or intermediate skin colour. This is expected since they were recent migrants from Africa and the Middle East. What is surprising is how long these ancestral variants persisted. Ancient genomes from European sites spanning the period from 40,000 years ago to around 8,000 years ago consistently show the ancestral dark skin variants in key pigmentation genes. The people who painted the caves of Lascaux and Altamira, who made the Venus figurines found across Ice Age Europe, who hunted mammoths and reindeer across the frozen continent during the last glacial maximum. These people likely had considerably darker skin than modern Europeans. The shift to lighter skin in European populations appears to be associated with two major demographic events. The first is the arrival of Neolithic farmers from the Middle East, beginning around 9,000 years ago. These farmers were already somewhat lighter than the hunter-gatherers they were moving in alongside. They had been living at slightly higher latitudes than equatorial Africa for several thousand years and may have already experienced some selection for lighter skin. The second and more dramatic event is the arrival of steppe pastoralists from the Eurasian grasslands, beginning around 5,000 years ago. These populations, who were linguistically the ancestors of most modern European language speakers, contributed significantly to the modern European gene pool and appear to have carried light skin variants at higher frequencies than either the hunter-gatherers or the early farmers. Professor Sven describes the population dynamics of ancient Europe as a series of waves, each one bringing new genetic material and reshuffling the frequencies of existing variants. The light-skin variants that are now nearly universal in European populations were not present at high frequency in any of the three founding populations of modern Europeans. They spread rapidly through all three after the populations mixed, because the selection pressure for lighter skin was operating on all of them equally. The mixing actually facilitated the spread of the light-skin variants, because it created a large, genetically diverse population in which a beneficial mutation could spread more rapidly than it could in a smaller, more isolated group. Modern Europeans are the genetic outcome of this mixing, a synthesis of three quite different ancestral populations, all of whom were subject to the same selective pressure for lighter skin in a northern agricultural environment. The agricultural revolution was not a single event, but a process that unfolded independently in at least 7 or 8 different regions of the world over a period of several thousand years. In the Fertile Crescent of the Middle East, wheat and barley were domesticated by around 10,000 years ago. In China, rice and millet were domesticated at roughly the same time. In New Guinea, root crops and bananas were being cultivated by 8,000 years ago. In Mexico, maize, squash, and beans were domesticated beginning around 7,000 years ago. In sub-Saharan Africa, sorghum and other grains were domesticated independently. Each of these regional revolutions involved the same basic transition, from a diverse, mobile, opportunistic diet to a more concentrated, sedentary diet based primarily on cultivated plants. The nutritional consequences of this transition have been studied extensively, and they are consistently negative in the short term. Archaeological evidence from the early agricultural period, across multiple regions, shows that populations becoming dependent on cultivated crops experienced reductions in stature, increases in dental disease, greater frequency of nutritional deficiency markers in bone, and higher rates of infectious disease. The diversity of the hunter-gatherer diet was replaced by the caloric density but nutritional narrowness of the agricultural diet. Vitamins and minerals that had been reliably obtained from a wide range of foods became scarce when the diet narrowed to a few staple crops. Vitamin D was not the only nutrient affected by the agricultural transition, but it was among the most severely affected for populations moving north. The dietary sources of vitamin D, fatty fish, animal organs, marine mammals, are not part of any agricultural staple. They are foods of the hunt and the coast, foods that sedentary farming communities in inland areas had limited access to. As communities settled onto their fields and built their villages and barns and granaries, they moved away from the coasts and rivers that had provided dietary vitamin D, and they moved toward a diet that provided essentially none. The consequences, as the skeletal record shows, were severe. Dr. Priya finds the agricultural revolution a useful lens for thinking about vitamin D in the modern world as well. The same dynamic, a diet that provides little vitamin D combined with limited sun exposure, has become characteristic of modern urban life. Most people living in cities, particularly in northern regions, spend the majority of their daylight hours indoors. Their diets, unless actively supplemented, contain little vitamin D. Their sun exposure is limited by clothing, indoor work, and the general structure of urban life. The result is widespread vitamin D insufficiency in modern populations, not as severe as in the early agricultural period, because modern food fortification and supplementation programs compensate to some degree, but real enough to have measurable effects on health. The Neolithic farmers' vitamin D crisis is being reenacted in the offices and apartments of the 21st century. The story of skin colour convergence in East Asian populations provides a fascinating parallel to the European story that also illuminates some important differences. East Asian populations developed lighter skin than equatorial Africans through a distinct set of genetic variants, operating through different molecular mechanisms. The most studied of these is a variant in the EDAR gene, which in addition to affecting skin pigmentation also influences hair thickness, breast tissue density, and the density of sweat glands. This pleiotropy, the fact that a single gene affects multiple traits, has made it more difficult to determine which trait or combination of traits was the primary target of selection when the EDAR variant spread through East Asian populations. Some researchers have proposed that the EDAR variant spread primarily because of its effect on hair thickness or on sweat gland density, which would affect thermal regulation in cold environments. On this hypothesis, skin lightening was a side effect rather than the primary driver of selection. Others argue that the vitamin D story is the most parsimonious explanation, since the same pressure was operating on East Asian populations as on European ones, and the same outcome, lighter skin, was produced. The debate has not been resolved, and it may be that different selective pressures were reinforcing each other, with the EDAR variant being favoured simultaneously for multiple reasons. The geographic distribution of skin colour in Asia is more complex than the simple latitude gradient that characterises Africa and Europe. South Asian populations, particularly those from the Indian subcontinent, show a wide range of skin tones that reflects the complex history of the region. Multiple waves of migration from different directions add mixture between populations of African, Middle Eastern, Central Asian, and East Asian ancestry, and diverse local environments ranging from tropical coasts to high mountain plateaus. The Tibetan population presents another interesting case. Living at extremely high altitude, they experience intense ultraviolet radiation despite their northern latitude, and they maintain relatively dark skin, consistent with the prediction that melanin levels track ultraviolet exposure rather than latitude directly. Dr. Amara has been involved in collaborative research with geneticists across Asia and Africa to map the full spectrum of skin colour variation in populations that have been relatively understudied in the genetic literature. Most of the early work on skin colour genetics focused on European and African populations, which created a Eurocentric picture that missed important complexity elsewhere. As more diverse populations are included in genomic studies, new variants are being discovered that contribute to pigmentation in populations across South Asia, Southeast Asia, and the Pacific. The full genetic architecture of skin colour in the global human population is considerably richer than the handful of well-characterised European variants that dominated the early literature. The trait that most complicates simple stories about northern adaptation is eye colour, and specifically the striking prevalence of blue eyes in northwestern European populations. Blue eyes are essentially absent in all other populations on Earth. They are vanishingly rare in Africa, uncommon in the Middle East, absent in East and South Asia, and largely absent in indigenous populations of the Americas and the Pacific. They are, in their geographic restriction, one of the most regionally specific traits in all of human biology. The genetic change responsible for blue eyes in European populations has been mapped to extraordinary precision. It is a single regulatory variant, not in a gene itself, but in a regulatory region near the OCA2 gene that influences how strongly OCA2 is expressed in the iris. The ancestral form of this regulatory region allows strong OCA2 expression, producing high melanin levels in the iris and dark eye colour. The derived form, found in virtually all blue-eyed people everywhere on Earth, reduces OCA2 expression in the iris, while leaving it intact in other tissues. The reduction in iris melanin produces the light scattering that appears blue. What makes this particularly striking is the evidence that all blue-eyed people in the world are descended from a single individual who carried the first copy of this regulatory variant. A single person, somewhere in or near what is now the Baltic region, perhaps 15 to 20,000 years ago, whose eyes were different from everyone around them. This is not a hypothesis, but a conclusion supported by genetic analysis. The blue eye variant is identical in all blue-eyed people everywhere, indicating a single origin. Every blue-eyed person on Earth, from Scandinavia to Argentina to Australia, is carrying the same mutation in the same regulatory region, descended from the same ancestor. Professor Sven finds this fact both humbling and slightly dizzying. The most distinctive visual characteristic of northwestern European populations, the feature that has been the subject of more art, more poetry, more cultural meaning than perhaps any other physical trait, traces to a single individual who lived before agriculture, before cities, before writing. Whether that individual was aware that their eyes were different from those of everyone around them, whether this difference was remarked upon or celebrated or feared, we cannot know. We know only that they had children, and their children had children, and down through 15,000 years of births and deaths and migrations and cultural transformations, that one regulatory variant persisted and spread until it now appears in the iris of hundreds of millions of people who will never know the name or the face of the person they have to thank for it. The health consequences of the mismatch between inherited skin colour and environmental ultraviolet radiation have become one of the more active areas of research in human physiology and public health, because the modern movement of populations has created this mismatch at an unprecedented scale. In the United Kingdom, for example, large communities of South Asian descent, many of whose ancestors came from regions of intense sunlight, have been living for generations at latitudes where winter sunlight cannot support vitamin D synthesis in any skin type. Studies consistently find high rates of vitamin D deficiency in these communities, with downstream effects that include elevated rates of rickets in children, osteoporosis in adults, higher susceptibility to certain infections, and possibly elevated rates of certain cancers. These are not trivial health problems. They are measurable, significant, and preventable. The irony, Dr. Priya notes, is that the communities most affected by this mismatch are often the least reached by the public health interventions designed to address it. Vitamin D supplementation recommendations and food fortification programs were designed with the health needs and cultural practices of the majority population in mind. They may not be equally accessible or effective for minority communities with different dietary patterns, different cultural attitudes towards supplementation, and different baseline risks. The population-level solution to a population-level problem created by the agricultural revolution, fortification of staple foods with vitamin D, works best when the staple foods in question are the ones that the population actually eats. At the other end of the mismatch spectrum, the experience of pale-skinned populations in high ultraviolet environments has been equally instructive. Australia provides what is perhaps the clearest natural experiment. Indigenous Australians developed dark skin over tens of thousands of years of living under intense Australian ultraviolet, skin that is well adapted to the radiation environment. European colonizers arrived with skin that had been shaped over millennia for the much gentler skies of northern Europe. The health consequences were immediately visible and have been extensively documented. Australia has some of the highest rates of skin cancer in the world, and the vast majority of this burden falls on the fair-skinned descendants of European colonizers. The adaptation that kept their ancestors healthy in northern Europe became a risk factor in the Australian sun. Modern medicine has developed extensive tools for managing this mismatch. Sunscreen with broad-spectrum UV protection protective clothing, behavioral guidelines, regular skin checks, vitamin D testing, and supplementation protocols. These tools are effective when used correctly and consistently, but they require knowledge, access, and motivation that are not equally distributed. And they are fundamentally responses to a problem that biology would have solved automatically, given enough time and enough generations through the same process that solved similar problems in the past. The question of whether human populations in new environments will eventually adapt genetically to their new solar conditions is one that researchers approach with caution. The timescales of genetic adaptation are long, the medical interventions are effective in the short term, and the global movement of populations means that no single equilibrium is likely to be established before conditions change again. The scientific story of skin color has important implications for how we think about human diversity more broadly, and these implications are worth articulating carefully. The variants that produce different skin colors are not representative of human genetic variation in general. They are variants in a small number of genes affecting a specific biological system, melanin production in the skin, that is under strong environmental selection. The selection is driven by ultraviolet radiation, which varies systematically with latitude and reflects the geography of where populations lived over the past tens of thousands of years. This geographic patterning gives skin color a systematic distribution across populations that makes it seem like a marker of deep biological differences. But this appearance is misleading. When geneticists look at the full complement of human genetic variation across all 20-odd thousand genes in the human genome, they find a very different picture from the one that skin color would suggest. Most human genetic diversity by standard measures exists within populations rather than between them. The differences between any two people drawn from the same population are, on average, larger than the differences between populations defined by continental ancestry or by the geographic categories that correspond loosely to what we call races. The visible traits that we use to classify people — skin color, hair texture, eye color, facial features — are among the most geographically differentiated aspects of human biology, because they are the traits under the strongest directional selection from local environments. They give the impression that human populations are more different from each other than they actually are at the level of the full genome. Dr. Amara uses a geographic analogy to illustrate this point. Imagine mapping the average temperature of cities around the world and finding that temperatures vary systematically with latitude. High temperatures near the equator, low temperatures near the poles. This geographic patterning would be real and significant for understanding climate. But it would be misleading to conclude from it that the cities near the equator and the cities near the poles were fundamentally different kinds of places. They have different climates, but they share the same basic urban infrastructure, the same range of human activities, the same fundamental character as human settlements. The climate difference is real. The difference in underlying nature is much smaller than the climate difference would suggest. The same logic applies to skin color and human genetic diversity. The geographic patterning of skin color is real and significant for understanding evolutionary history, but it should not be mistaken for a guide to overall genetic similarity or difference. The deepest implication of the skin color story is a temporal one. The genetic variants that produce different skin tones are recent, evolutionary recent, meaning on the order of tens of thousands of years rather than millions. For the overwhelming majority of our species' existence, for the first 90% or more of our 200,000-year history, all human beings had dark skin. The variation that now seems so fundamental and so defining was not there. It arrived late, it arrived through specific and understandable mechanisms, and it arrived in service of a specific and understandable biological function. Understanding this changes the relationship between skin color and human identity. Skin color is not where the human story begins. It is a recent annotation on a much older and more continuous text. The long-term consequences of the agricultural transition for human biology extend well beyond vitamin D and skin color. The shift to grain-based diets also changed the selection pressure on digestive enzymes, on immune system genes, on the microbiome, and on a range of other physiological systems that had been calibrated to a hunter-gatherer way of life. The human body as we experience it today, with all its characteristic vulnerabilities to modern disease, its complex relationship with food, its seasonal fluctuations in immune function, is in many ways the body of an agricultural species that is still in the process of adapting to a way of life that is only 10,000 years old. In evolutionary terms, we are recent converts to farming, and many of our health challenges are legible as the consequences of that conversion. Professor Sven has become interested in what he calls the archaeology of health, the reading of ancient skeletal remains not just for demographic information, but for evidence of the physiological challenges that populations faced at different periods. The transition from hunter-gatherer to agricultural communities is visible in the bones. Average stature decreases. Dental wear patterns change from the broad, multi-directional wear of people chewing diverse foods to the more specialized wear of people grinding grains. Markers of nutritional deficiency, evidence of periods of growth disruption visible as lines in the enamel of teeth, become more common. The agricultural revolution was a civilizational triumph and a biological stress test simultaneously, and the bodies of the people who lived through it bear the marks of both. Technology has not only allowed people to live under skies their skin was not calibrated for, it has also begun to alter the evolutionary dynamics of skin color in ways that are genuinely novel. When the severity of vitamin D deficiency was routinely fatal, as it was in early agricultural populations, the selection pressure on skin pigmentation genes was intense. When medicine eliminates the fatal consequences of vitamin D deficiency through supplementation and treatment, the selection pressure is reduced. Natural selection can only act on traits that affect survival and reproduction, and if a trait no longer affects either of these things, because its health consequences are managed medically, then selection on that trait weakens or disappears. The implication is not that skin color will stop changing. Other selection pressures, including ultraviolet-related skin cancer risk and whatever selection pressures we have not yet identified, continue to operate. But the specific pressure that drove the most rapid changes in skin color over the past 10,000 years, the fatal consequences of vitamin D deficiency in agricultural populations at northern latitudes, has been substantially reduced by the combination of food fortification, medical supplementation, and improved understanding of vitamin D requirements. We have, in a sense, solved the problem that skin color was evolving to solve. The question of what this means for the long-term trajectory of human pigmentation is one that is genuinely open. There is something philosophically interesting about the fact that the trait around which so much human social architecture has been built, the trait that has been used to justify slavery and colonization and countless other forms of systematic oppression, is, from the perspective of evolutionary biology, one of the most superficial and recently evolved aspects of human biology. Not superficial in the sense of trivial, the health consequences of the skin-sunlight interaction are real and significant, but superficial in the sense of involving only the outermost layer of the body, controlled by a handful of genes, evolved in response to a straightforward environmental variable, with no implications for any of the cognitive, behavioral, or social capacities that actually determine what a human being can do and be and become. The evolutionary story of skin color is in this sense an argument against taking skin color seriously as a marker of anything beyond what it actually is. A record of ultraviolet exposure in ancestral populations, it is an argument made not by philosophers or political theorists, but by the data itself, by the genetic evidence that skin color variants are recent and superficial, and environmentally responsive and shared in complex ways across populations that our social categories would treat as distinct. The data does not tell us what to do with this understanding, but it does clarify what skin color is and what it is not. And that clarification, pursued with patience and honesty through the tools of modern genetics, seems like a contribution to human understanding that is worth the effort of following all the way to the end. The three scientists whose perspectives have guided us through this story are each working on a version of the same question from a different angle. Dr. Amara is asking about the genetics, which variants, in which genes, at which frequencies, in which populations, at which times. Professor Sven is asking about the history. When did the changes happen? Who were the people involved? How does the genetic story map onto the archaeological and demographic record of actual human populations? Dr. Priya is asking about the physiology. What are the health consequences of this system for people living today in environments that may or may not match their evolutionary heritage, and what can medicine and public health do about the mismatches? These three questions are not independent. The genetics informs the history and the physiology. The physiology explains the selection pressure that drove the genetics. The history contextualizes both. Together they constitute something that feels genuinely new in human self-understanding. A scientific account of one of the most visible aspects of human variation that is specific enough to be meaningful, honest enough to acknowledge uncertainty, and complete enough to place the variation in its proper evolutionary context. It is an account that replaces mystery and mythology with something more accurate and ultimately more interesting. Not less wonderful, the story of how a single mutation in a single gene, arising in a single individual on the shores of the Baltic Sea 15,000 years ago, eventually spread to populate the eyes of hundreds of millions of people, is as wonderful as any mythology. But the wonder is grounded in what actually happened rather than in what we wished, or feared, or imagined. What you carry tonight as you settle towards sleep is the end of a chain that reaches back through 100,000 generations to people who stood in the open air of an African morning and felt the sun on their dark skin and went about the work of surviving. Every generation between them and you added something. A mutation here, a migration there, a dietary change, a seasonal adaptation, an agricultural revolution, a ship crossing an ocean. All of it accumulated in the person you are, in the skin you are wearing, in the genes that produced it. The story is not finished. Your descendants will add their own chapters, but tonight's chapter at least is complete. This has been Midnight Chronicle. Sleep well wherever in the world you are tonight.